Good evening, ladies and gentlemen. We're back once again with another amazing science tutorial video. I'm Coach Spivey, joined with my son,
Jordan Spivey.
And today we're going to be teaching you about protein synthesis. So let's begin. So what is DNA? DNA is the blueprint of life. It has the instructions for making an organism. A gene is a segment of DNA that codes for a protein which determines a trait, such as skin color or eye color. If you notice, if you look at me and my son, a lot of people say we look awfully alike. So we have certain genes or traits that are very much in common. And DNA is wrapped around a protein to form a chromosome. So the structure of DNA was discovered by James Watson and Francis Crick in 1953. And they described it as a double helix or a twisted ladder. And if you look at over here to the right, here's that structure of DNA. If you notice, you have these different colors because the red and the greens match with each other and the yellow and the purples match with each other. And we'll be covering why these colors or these bases match with each other in the upcoming slides. So those colors we were talking about earlier are the nitrogen bases. And DNA has 4 nitrogen bases. We have adenine, thymine, guanine, and cytosine. And they're attached to a deoxyribose sugar. And they always follows the base pair rule. So that means that adenine is always going to pair with thymine and guanine is always going to pair with cytosine. And they pair up as follows. A goes with T and C goes with G. And they're connected by weak hydrogen bonds. So let's look at our first check for understanding. You have 1 minute to complete and answer the following questions, and I'll pause that video and give you that 1 minute starting now. Alright, so let's take a look at our first check for understanding. So Jordan, number 1, what is the major purpose or function of DNA?
The major purpose/function of DNA contains the instructions for making an organism.
Good. Number 2, what are the 4 nitrogen bases in DNA?
Adenine, thymine, guanine, and cytosine.
Very good. And number 3, how do the nitrogen bases pair with one another?
Adenine goes to thymine, and then cytosine goes to guanine.
Very good. And then number 4, who discovered the structure of DNA and what did they describe it as?
James Watson and Francis Crick discovered the structure of DNA, and he also said that it was like a double helix, aka twisted ladder.
Alright, very good, Jay. And you get a crisp high five. Alright, let's move on to our next bit of material. So let's take a look at it. So we said earlier A goes with T and C goes with G. So if you take a look, if we have A right here, then T would be right here. A, C, G, C. And on this other side, we will have A, T, T, G, C, G. Does that make sense, Jay?
Yes, it does.
Alright, so let's take a look and see if we was correct. Yay, we was correct. Good. And then if you notice, 2 identical strands of DNA are formed before the cell goes through mitosis so that each new cell has the same DNA. And if you take a look at this side right here. This left side is completely identical to this left side. Same thing with this right side of this DNA strand. Completely identical to this right side of the DNA strand. So this type of replication is called semi-conservative because there is half of the original strand is in each DNA copy. So now let's take a look at protein synthesis. And the reason why protein synthesis starts with DNA but doesn't end with DNA because DNA is too big to leave the nucleus. So RNA or ribonucleic acid must get involved. So there are certain similarities and differences between DNA and RNA. Both have the nitrogenous bases adenine, guanine, and cytosine, and phosphates. But the differences between the two, DNA is deoxyribose sugar, while RNA is ribose sugar. DNA contains thymine, while RNA contains uracil instead of thymine. And then DNA is double-stranded, and RNA is single-stranded. So now for your second check for understanding, you're going to make a Venn diagram to show the similarities and differences between DNA and RNA. I give you 2 minutes to set up and make your Venn diagram for DNA and RNA, and we'll start those 2 minutes now. We're back once again, ladies and gentlemen, and let's go ahead and check and see how you did on your second check for understanding. So Jordan, what is the difference between DNA and RNA?
DNA has deoxyribose sugar, and RNA has ribose sugar. And then DNA has thymine, and RNA has uracil. And then DNA has double-stranded, and then RNA is single-stranded.
Good. And what do they have in common?
They both have adenine, and then guanine, cytosine, and phosphate.
Very good, sir. All right, let's continue with our material. So there are three types of RNA. We have mRNA, which is messenger RNA, rRNA, which is ribosomal RNA, and then we have tRNA, which is transfer RNA. So the mRNA is responsible for making a copy of the DNA code and taking it to the ribosome, and this process is called transcription. And then rRNA actually makes up the ribosome, so that's why we call it rRNA or ribosomal RNA. And then transfer RNA or tRNA picks up an amino acid in the cytoplasm and it actually brings it to the ribosome. So there are two steps for making a protein. Step number one is transcription, and that's when DNA is copied by mRNA in the nucleus. So if you take a look right here, here's our DNA strand with this double helix, and here it's located in this cell nucleus. And then mRNA makes a copy of that DNA strand, or a copy of that DNA strand in transcription, and takes it to the ribosome. So let's take a closer look at transcription. And we stated earlier that transcription occurs in the nucleus during interphase of the cell cycle. If you notice right here, we have the DNA, and that DNA is going to be transcribed into mRNA, like so. And remember, RNA has uracil instead of thymine to bond with adenine. And then mRNA molecule will exit the nucleus and go to a ribosome in the cytoplasm. So step two of protein synthesis is translation. And tRNA picks up the correct amino acid in the cytoplasm, and then it's going to take it to the ribosome to make the protein coded by mRNA. So if you notice, let's backtrack. We have the DNA in the nucleus. The DNA is transcribed by the mRNA. And the mRNA goes in the cytoplasm and attaches to the ribosome. And then we have tRNA that picks up an amino acid out of the cytoplasm and brings it back to the ribosome. The mRNA molecule codes for a protein during translation. So this occurs in the cytoplasm on the ribosome. And each group of 3 mRNA bases, which is a triplet codon, or we call it a codon, codes for 1 amino acid. So for example, if we got a triplet codon of ACU, if we use this codon wheel right here, we would start in the middle and then work our way out. So we would start with A, C, U, and that would let us know that it's the amino acid threonine or threonine. And then say for another example, say we had GAC. And then we would take this and we would look at GAC. Notice how we start in the middle and then work our way out, and that would let us know that the amino acid would be aspartic acid. So there are 20 amino acids that exist in all, and amino acids are carried to ribosome by the tRNA with the anticodon, and amino acids bond together to form proteins or polypeptides. So now it's time for your third check for understanding. You'll have 2 minutes to answer the following questions, and we'll give you those 2 minutes beginning now.
Alright guys, now it's time to check our third check for understanding. Alright, so Dad, what are the 3 types of RNA? What are their functions?
So the 3 types of RNA are mRNA, which mRNA transcribes DNA and takes that message to the ribosome. And then we have rRNA, and it makes up the ribosome. And then finally we have tRNA, and it transfers amino acids from the cytoplasm to ribosomes.
Alright, very good. So now for our second question. Write the transcripts mRNA of the DNA molecule below.
Okay, so what I like to do on mine, I always like to separate mine in 3s. Remember, those codons come in 3s. This also helps me to organize my information when I write it. So ATG transcribes to UAC, GAC transcribes to CUG, TAC to AUG, and then GCA to CGU. Remember, when we go from DNA to RNA, that thymine turns into uracil.
Alright, so now for our third question. Where are proteins made?
Proteins are made in the ribosomes.
Alright, now for our fourth question. What are the building blocks of proteins?
And everyone knows those are amino acids.
Yes. And we have a good old fist bump.
All right, let's move on to our next material. So let's do a review, everyone. The code for making the protein must be copied from the DNA and taken to the ribosome. So the DNA gets transcribed into mRNA. Then this mRNA leaves the nucleus, goes to the cytoplasm, and attaches to the ribosome. Then amino acids must be put in the right order at the ribosome to make the right protein. So this is the job of the tRNA. And these are the jobs of both the mRNA and tRNA. So let's take a look. What amino acids would the following mRNA codons code for? So we used the codon wheel earlier, but now let's go ahead and use the codon wheel and codon chart to show you how easy it is to find the code for the correct amino acids. And I'll go ahead and change the pen color to red so you can see it. So this right here is our codon charts over here. And this is our codon wheel. You can use both to find the right amino acids. It just depends on your personal preference. Some people like the codon chart and some people like the codon wheel. So let's take a look at our first codon. We have AUG. If we look on our codon chart, here's A, here's U, and then here's G. So we start off with our first base, here's our second base up top, and here's our third base. And we cross all three of those where they meet it. So we have A right here, U, and then we come down here, and that codes, and then we have G, and it codes for methionine. And if you look on your codon wheel, we start off in the middle right here, and then we start off, and then we work our way out. So we have A, U, G, methionine. Like I said earlier, both of them do the same thing. It just depends on your personal preference. So let's look at our second one, CCG. And let's use our codon wheel this time. So we start with C, work our way out, C, and then to G. And that codes for proline. If you notice on our codon chart, C, C, and then G, proline, the same thing. And then let's look at our third one, AUU. And let's look at our codon chart this time. So we start with A, U, and then we take this U across, and that will code for isolene. And we look A, U, U. And I apologize, it should be isoleucine instead of isolene. And then let's look at our fourth one. We have UGA. Let's use our codon wheel for this one. UGA. And that would be our stop codon. Now it's time for your fourth check for understanding. And given the following DNA sequence, give the mRNA sequence and the amino acids produced. You can use either the codon chart to help you with your amino acids or the codon wheel, whichever one you're most comfortable with. You have 2 minutes to do so, and we'll begin those 2 minutes starting now. So now let's see how you did on your fourth check for understanding. And we have the DNA codon right here, GCC. What mRNA codon does this code for, Jordan?
G goes to C.
Uh-huh.
And then C goes to G.
Yes, sir.
And then the other C goes to G.
All right, very good. What about this one?
A goes with U, and then A goes with U again, and then T goes with A.
Good. And what about TCG?
A, G, C.
Good.
And then A, C, U.
All right, very good. So now let's take a look at the amino acids that code for these codons. So CGG. So let's take a look at our codon wheel. CGG. We have arginine. So ARG. And then we have UUA. And let's look at our codon chart. So UUA. And that's going to be leucine. Then we have AGC. So AGC. We have serine. And then we have ACU. So ACU. And we have threonine. I know you did very well on this. Good job. Fist bump. So now let's take a look at the big picture. So we have DNA, which is in the nucleus, and it gets transcribed by mRNA. The mRNA leaves the nucleus, goes to the cytoplasm, and attaches to the ribosome. And the mRNA is translated into making a protein once the tRNA brings an amino acid from that cytoplasm. So the first part is transcription. We have DNA and it's copied into mRNA in the nucleus. The mRNA leaves the nucleus. Then step 2, translation. The tRNA picks up the correct amino acid in the cytoplasm and takes it to the ribosome to make the protein coded for by the mRNA. Once again, we have the DNA in the nucleus. The DNA is transcribed by mRNA. The mRNA leaves the nucleus, travels in the cytoplasm, and attaches to this ribosome. And then tRNA picks up an amino acid out of the cytoplasm and attaches in the correct order to the ribosome and helps and works with mRNA to get that code to make the correct amino acid to make that protein chain. So here's another look at protein synthesis. So let's look at this overview. So first part, transcription. Transcription starts in the nucleus, and we have that DNA, which is transcribed by mRNA, and the mRNA makes those codons for proteins. And then step 2, we have translation, which occurs at the ribosome. So we have the mRNA with the codons. The tRNA picks up an amino acid and has the anticodons, and they match up at the ribosome. And then those amino acids help to make a protein. Let's do an overview of protein synthesis. And protein synthesis is the making of a protein. And in order to make a protein, a cell needs the following big time players: DNA, which holds the code in the nucleus; mRNA, codons, which carries the code to the ribosome; then ribosomes, where the actual protein is made; and then tRNA, which is the anticodon, which picks up an amino acid out of the cytoplasm and brings them to the ribosome. And then we have amino acids, which actually are the building blocks of a protein. So now it's time for your 5th check for understanding. So you'll have 2 minutes to complete the following information, and we'll begin those 2 minutes starting now.
Alright, we are back. Now it is time for our 5th check for understanding. Protein synthesis is the making of a protein. To make a protein, a cell needs DNA, which holds the code in the nucleus; mRNA, which carries the code to ribosome, where protein is made; tRNA picks up amino acids and brings them to ribosome. Amino acids, building blocks of proteins.
Good. High five. So now let's do a summative review of everything you've learned in this video. So we're going to use the following diagram above to answer these questions below. So number 1, from which labeled structure is structure D made? If you notice in structure D, we have AUG. And if you look up top, we have TAC. This T goes with this A goes with this U, and this C goes with this G. This information is coming from the DNA that's located in the nucleus. So this DNA is being transcribed into messenger RNA right here at point D, or structure D. And then it says identify that labeled structure. That labeled structure is DNA. So point A is DNA. Number 2, identify structure F. Structure F comes in 3, so this lets me know that this is a codon. And then what does it code for? We're going to use our codon wheel to see what it codes for. So we have GCU, and let's start from the middle, GCU. So this actually codes for alanine. Then number 3, what is structure E and what does it specify? Structure 3 comes in E's as well, but structure 3 starts at the very beginning of this mRNA strand. So this is actually a start codon, which starts the process of making an amino acid or to make proteins. So now let's go ahead and take a look. AUG is actually methionine, and it specifies to begin or to start making amino acid or protein. So start making protein. And I'll abbreviate that. Then number 4, what would happen to structure F if structure C were deleted? So structure C is one base pair right here, one base, and if structure C was deleted, that means that this codon would come up short. So there will be a deletion. So that would actually be considered a deletion, or which would lead to a mutation. And number 5, what processes do X and Y represent? Well, here's X right here, and this X represents transcription, because we have this DNA that's being transcribed into mRNA so it can leave the nucleus. And then if we take a look at structure Y, this is translation. So we have that mRNA being translated to make an amino acid to make protein. And number 6, what does this entire diagram represent? This entire diagram represents protein synthesis, starting off with our DNA and then going through transcription, going to mRNA, and then the mRNA being translated into amino acids to make proteins. Ladies and gentlemen, I'm Travis Spivey signing off with my son,
Jordan Spivey.
And I hope this tutorial video was helpful. Peace, and y'all have a wonderful, awesome day.